Today, we are going to cover Chapter 8, which deals with the basis of reproduction and inheritance in our body's cells. So this deals with a process called mitosis, which is the division of our body cells, our somatic cells, and then also we'll talk about meiosis, which is the division of our sex cells. Our sex cells are also known as gametes, and gametes are our sperm and egg. So let's get started. All right, so let's look at 4 of the overarching big ideas from this chapter. We're going to talk about cell division and reproduction, the eukaryotic cell cycle and mitosis, meiosis and crossing over, and then alterations of chromosome number and structure, and then what type of organisms are produced from those different combinations. All right, so let's start off with cell division and reproduction. So cell division, this is a very important context or a very important concept in biology. So cell division is at the heart of reproduction. It can either be sexual, so think of how humans and animals reproduce, or it can be asexual. And asexual reproduction happens mostly with bacteria, some other plants, other organisms as well, but whereby a cell just divides and splits and makes an exact copy of itself, but it does not need any kind of external factors in order to actually divide, or we can say reproduce. Okay, so looking at our little starfish, so we all pretty much know if you cut off an arm of a starfish, it can grow a whole new part of itself or a whole new organisms. If anyone likes plants, you know, if you cut maybe a cutting sometimes or a leaf, you can just plant that and grow a whole new plant. And then, of course, we as humans use sexual reproduction to produce more of our offspring. All right, so the way that prokaryotic cells reproduce asexually is something called binary fission. Okay, and I'll put binary. So we know that bi means 2, right? So kind of think of dividing into 2 or just dividing in half. So in prokaryotes, their DNA is a little bit different. It's just kind of one big circle called a plasmid. Okay, so the cell replicates its single chromosome. The copies move apart to separate poles or opposite poles of the cell. The plasma membrane then pinches inward, and the more a cell wall is then eventually made and surrounds the 2 new daughter cells. Let's take a look at how that occurs. Oops. Here we go. So first off, you see we have a prokaryotic cell. So think of our things like bacteria, archaea. You just have one cell right here, and here is that kind of rolled up circular chromosome. The chromosome or DNA duplicates. It moves to opposite sides of the cell. You get it kind of pinching in between the 2 cells. And then now you get a new cell wall formed around the 2 new daughter cells. Okay, so just kind of dividing in half. So kind of the same concept with these eukaryotic cells. We're going to talk about something called mitosis, and this is the way that our body's cells divide. All right, because eukaryotic organisms are typically much more complex than anything that is prokaryotic, there are, of course, a lot more regulations and controls on how our cells divide. So our genes are grouped into multiple chromosomes in the nucleus. So each chromosome contains one long DNA molecule. It's a little hard to see individual chromosomes under a microscope unless they're actively dividing. That is the part when the chromosomes actually condense and become more visible. Okay, so as the chromosomes condense, they become something called chromatin. So these are very, very tightly packed together, and they're bound very, very tightly with something called histones. And these are more of like a stabilizing protein that keep that DNA tightly wrapped up. Okay, so before a cell starts dividing, the chromosomes duplicate, producing something called sister chromatids. And these sisters actually contain identical DNA. So first, this is just one chromatid. It looks like a little leg, right? And then it replicates and duplicates, and now it has a little sister. Okay, and these both have the exact same type of DNA. It's going to be identical. In the middle of these two sister chromatids, we have something called a centromere. Okay, and this kind of holds these two sister chromatids together. All right, so kind of looking here, we can say this is an example of maybe interphase or prophase. The DNA or chromatid, you can see right here in the middle of the cell where the nucleus would be. Okay, so this is just one chromatid by itself. When we have two together, these are sister chromatids joined in the middle by something called a centromere. After cell division, these sister chromatids are pulled apart to opposite sides of the cell so that each new cell that's being made gets an exact copy of DNA. All right, so a major part of a cell's life cycle is something called the cell cycle. This is kind of an ordered sequence of events that kind of extends from when the cell is first formed to when it starts actively dividing. So the cell cycle, it takes about 23 hours to kind of rest, grow, and replicate DNA in something called interphase. Interphase is actually the longest part of the cell cycle. It takes about 23 hours. Okay, so kind of resting and growing. So we have our G1 phase, our S phase, and our G2 phase. Think of G meaning growth and S for synthesis. So the DNA is making copies of itself. All right, and then we have the mitotic phase, and this is the actual active dividing of the cell. This takes about 1 hour, okay? So you can kind of see the cell is mostly resting and growing and making more copies of DNA for that longer period of time and only actively dividing for about 1 hour in that cell cycle. All right, so mitosis distributes duplicated chromosomes into two daughter nuclei. So we start out with one mother cell, and it produces two exact clones of itself. Okay, so 1 and 2. So let's take a look at what happens during the different parts of the cell cycle. This big purple blob you see right here, this is the nucleus. We have the nuclear envelope surrounding it, and within the cell, you can see all of this chromatin, which is our DNA. All right, this is the first part of mitosis, which is that cell division. This first part is something called prophase. So you can see here that the nuclear envelope starts to disintegrate. The nucleolus disappears, the chromatin starts to condense, and then our spindle, which is consisting of something called centrosomes, our spindle, which is actually the part of the cell, something called the cytoskeleton, that's going to help to pull the chromatin apart, okay? So kind of early prophase and late prophase right here. You have the larger spindle formation. Later on, you're going to have the spindles. You see how they're attaching, and you kind of see something right here, how they're attaching to that centromere, one on each side of the cell. So when these spindles actually contract, they kind of pull the sisters apart, and so that each new cell is going to have an exact copy of DNA. Okay, this stage of mitosis is called metaphase. So think of things lining up on the middle of the cell. So this is when the chromatids, the sister chromatids, line up on the metaphase plate in the middle of the cell, and you have the spindle attaching to one side of the centromere for each sister chromatid. That's metaphase. Next, we have anaphase. So this is when the spindle contracts and pulls the sisters apart to opposite poles of the cell. And you see here how we just have these one little legs being pulled over. So now the sisters have been pulled apart. And so now in this last phase, we kind of combine something called telophase and cytokinesis. Telophase. This is when you actually get the equal division of DNA to opposite poles of the cell, okay? So the nuclear envelope starts to reform. The chromatin kind of gets less dense, more diffuse, and the spindle starts to go away. We get something called a cleavage furrow. This is in animal cells where you get a cleavage furrow. And then the cell is going to keep going, keep dividing here until they pinch off, and you get 1, 2 new exact copies of that original mother cell. Okay, kind of combining telophase and cytokinesis. So telophase is the equal distribution of DNA contents, while cytokinesis is the equal division of cellular content, so things including the cytoplasm and the organelles. If we are talking about plants rather than animals, they don't have a cleavage furrow. They form a cell wall in between. So looking at this, they kind of go through the whole cycle, just like with animal cells. Only you see here, we start having a cell plate formation right here and no cleavage furrow. The cell plate just keeps growing towards the ends of the cell until we actually get a new plate formed. And now we have 2 separate daughter cells that are exact copies of that mother cell. Okay, so within the cell cycle, we have a few controls. So proteins that either tell cell division to go forward or proteins that actually stop cell division. So when these different proteins can be mutated, and if they are, they actually kind of promote cancer formation because those controls are no longer working to say, hey, we have enough, let's stop the cell cycle. These opposite controls that are, when mutated, they just tell the cell to keep dividing and dividing and dividing until you get that first initial tumor, and then it can metastasize or spread throughout the body from there. Okay, so we have a few checkpoints. So the first one is called G0, so our growth phase 0. We have a G1 checkpoint. So all these checkpoints are here to check the DNA. So if there's anything wrong, any kind of mutation, the DNA and the cell itself gets destroyed, okay? Because if something is mutated, if the DNA is having any kind of mutation within it, that just kind of increases the chances for cancer formation. And this is why these checkpoints are so important. All right, so cancer is defined as the uncontrolled proliferation, which is also the division of cells. And when this happens, we get a tumor, right? And then if the tumor cells spread, they become malignant, okay? If they are malignant cells, they kind of invade other tissues, they invade the lymph nodes, they get into the blood supply and the blood vessels, and can therefore spread throughout the body. All right, so the major treatments nowadays, of course, radiation, chemo, and then certain types of cancers do have drugs targeted to their destruction. Okay, so for example, we have a type of breast cancer. So we have maybe stage 1 cancer where we have tumor formation. We have the spreading of that kind of cancer. We can say this is stage 2. It'll spread to other organs in stage 3, and then stage 4, it has entered the lymph vessels and the blood vessels and can therefore spread to any part of the body. If it is breast cancer, it naturally spreads first to the lungs and then can travel because it's so close to the neck blood vessels, it can travel to the brain very easily. So those are 2 of the top places where breast cancer metastasizes. Okay, so mortality rates from cancer vary by age, diagnosis, race, and other factors, of course. So for example, the risk of breast cancer does increase with age, but you kind of see here that Black women, we'll say women, just because they have a higher mammary gland kind of concentration than males. So we'll say Black women. And then you see here where it says below age 35. So we can say that premenopausal Black women that are typically very young usually are diagnosed with an advanced breast cancer and kind of a higher mortality than any other race that we see here on this chart. All right, so let's talk about meiosis and crossing over. So meiosis is the division of sex cells. Sex cells are gametes. And if you remember from the beginning of the lecture, we talked about sperm and eggs. So our body cells are also known as somatic cells. So these are the types of cells you find in your hair, your liver, your heart, just our basic body cells. So for human cells, we have 46 chromosomes and 23 pairs. Okay, so these chromosomes carry something called homologous genes. Okay, so it carries kind of copies for each gene, right? So one on each chromosome. And this helps with genetic variation. And also if one chromosome is affected with some type of genetic disease, there is another healthy chromosome there to kind of take the place for it. Okay, so here are our chromosomes. Of course, they're duplicated. So we have our two sisters. We have our centromere right here. Think of the center, right? Holding the two together. Okay, and then they are duplicated. So looking at the different colors, you see here we have pink, we have yellow, we have green and blue. All of these represent different genes, something called an allele, that code for very similar characteristics. Okay, so cells with two sets of homologous chromosomes are something called diploid. So these are going to be our somatic cells, and they are going to have 46 chromosomes. Okay, so 46 chromosomes is also diploid, and this is our 2n number. Okay, so on the other hand, our gametes, which are the eggs and sperm, these are haploid cells. So they only have 23 chromosomes, and this is just your basic N number. Okay, so kind of think about that, why eggs and sperm would only have 23 chromosomes each. It's because during fertilization, when the sperm joins and fuses with the egg, the new forming fetus is going to get one set of chromosomes from the mother and one set from the father to equal out to 46 chromosomes. Okay. So kind of just looking through here. So meiosis is very similar to mitosis. There are a few key differences though. So for meiosis, it actually goes through 2 cycles of division. Okay. And at the end, you're going to get typically 4 haploid cells that are non-identical. And this is a big thing to remember because with mitosis, the cells that were formed were identical, right? So with meiosis, we want to get these non-identical cells, which increases genetic variation, which therefore increases the survival of the species. So first, we have meiosis 1. Okay, so you get the formation of 2 cells, and then meiosis 2, where those 2 cells further divide into those 4 new cells. This is most common for sperm cells. With egg cells, it's a little bit different. Okay, so meiosis, of course, it's preceded by chromosome duplication, but in meiosis, of course, the cell is going to divide twice to form 4, and I would always put non-identical, okay? So the first division of meiosis 1 starts with the pairing of homologous chromosomes. So remember here and here, we have 1 chromosome, so 2 sister chromatids, and then we have another pair lining up right next to it. So they call these homologs. Okay, so these are homologous chromosomes. They also call them tetrads because tetra means 4, right? Tetrads. And you can see here that we have 4 legs, right? 1, 2, 3, and 4. Okay, so meiosis 1 consists of these homologous chromosomes lining up together, and then just the pairs being pulled apart to opposite sides of the cell. Okay, so you see here we have our homologous chromosomes lining up together during metaphase 1. But then during anaphase 1, you see we're just having those pairs pulled apart. We're not pulling apart the sister chromatids just yet. Okay. During the phase kind of between meiosis 1 and meiosis 2, there is no more DNA replication. So after this, meiosis 2 is the most identical to mitosis. You see here we have the sister chromatids lining up on the metaphase plate. And then anaphase, you're actually pulling apart the sister chromatids, okay? So that one's very, very close to mitosis. Okay, so with mitosis, you're going to get 2 genetically identical diploid somatic daughter cells, okay? So with meiosis, though, you're going to get 4 genetically unique haploid gametes. Okay, so there's a little comparison chart if you want to take a look at that. All right, so independent orientation of chromosomes in meiosis and random fertilization lead to varied offspring. Independent orientation, same thing as independent assortment. So each chromosome of a homologous pair differs at many points, of course, from the other member of the pair. So this random arrangement of chromosome pairs at metaphase 1 of meiosis leads to many different combinations of chromosomes in eggs and sperm. And then, of course, the random fertilization of eggs by just one sperm greatly increases this variation. Okay, so let's take a look. Kind of looking here, you can see all the different possibilities. Here's possibility A, possibility B. You can see how they're kind of lined up differently. Just random assortment of how we're going to get this new DNA formation. All right, so the differences between homologous chromosomes come from the fact that they can bear different versions of genes at corresponding loci, and these are called alleles. So think of genes for height, for hair color, all those different things. You're going to have 2 alleles or 2 genes coding for each thing. All right, so during prophase 1 of meiosis, you're going to get something called crossing over. And this is kind of when homologous pairs, they're sitting next to each other, and then they start exchanging pieces of themselves. So let's say we have 1 homologous chromosome here and 1 right here. And because they're so close together, they can kind of swap out pieces of themselves and pass it to the other chromosome. And now you have a new combination or a recombination of genes. Okay, so just kind of looking at these 2 mice, they're going to have different fur color, different eye color, of course. And so they're going to get different alleles that actually code for these different traits. Okay. So looking at this, we have something called a chiasma forming, and this is kind of when you get that crossing over. See how now this red sister chromatid now has a blue piece, and how a piece of this blue sister chromatid now has a red piece because they exchange that DNA, those alleles during crossing over. Okay, still looking at different types of things that happen. I would go through these charts to kind of get a better idea. I know it's kind of small right here. All right, so last part of chapter 8, these are alterations of chromosome number and structure. These can be due to a variety of things, but most of these can result in some type of disease or syndrome. So an abnormal chromosome count is the result of something called non-disjunction. And so this can happen in a couple of different ways. It can result from the failure of a pair of homologous chromosomes to separate during meiosis 1. So instead of those homologs being pulled apart to opposite sides of the cell, they remain joined together. Or it can be the failure of sister chromatids to separate during meiosis 2. So it can occur in either the first or second part of meiosis. So let's take a look. So if we have non-disjunction here in meiosis 1, and we have this homologous pair, they did not get pulled apart. You see with the formation of the 2 new cells, we're going to get more than the normal amount of chromatids and DNA in 2 of the 4 gametes, and then not enough chromatids or DNA, of course, in the other 2. All right, so let's say now we have a normal meiosis 1, but then we have non-disjunction during meiosis 2. So occurring right here where the sister chromatids are not fully pulled apart. So you get a lesser amount in one gamete, and then you get a higher amount in the other gametes. So for instance, let's take a look at something called a karyotype. So looking at this, we have our full complement of chromosomes. We have our sister chromatids here and here, and our centromere holding them together, okay? So these are lined up as homologs, okay? So these are homologous chromosomes in our karyotype, and it tells doctors and scientists whether there is any kind of visible type of either multiplication or reduction in chromosome number. So we have, you see our 23 pairs of chromosomes with that last or 23rd pair representing the sex of an individual. So an XY, this will be a male, and an XX would be a female. Okay, so there is a syndrome called trisomy 21, also known as Down syndrome. This is very, very common. Okay, so this means at the chromosome 21, there are going to be 3 copies of that chromosome instead of just 2. And that's why they call it trisomy. So 3 of that 21 chromosome. And there are a few very, very common features for this syndrome. Okay. Some of it can be attributed to an older age of the mother. So increasing, of course, after age 35 to on up. So that's why there's so many tests given to women, especially women who they consider to be an older age, so after age 35. But they ask if you do want to have these tests taken. All right, so non-disjunction of sex chromosomes during meiosis can result in individuals with a missing or an extra X or Y chromosome. So in some cases, so let's say an XXY, this leads to syndromes that can affect the health of the individual. In other cases, so XXX, the body is normal. So you're going to have 3 X's, and this person appears as female. Okay, so here's a little chart just detailing the different types of syndromes. So an XXY is something called Klinefelter syndrome. This is considered a genetic male. Okay, so this type of person will be sterile, underdeveloped testes. Sometimes they will have secondary female characteristics. An XYY, no big symptoms, okay? A triple X, okay? So maybe slightly taller. An X and an O, okay? So this is something called Turner syndrome, and this is considered a genetic female. So this person will also be sterile and have immature sex organs. All right, so that is the end of chapter 8. I do encourage you to do more of your own research, especially into the different syndromes, the different diseases, cancer that we talked about. They can be very, very informative. And don't forget to take your chapter 8 quiz. All right, see you all next time.